Welcome to a new IPOPI webinar on PID and vaccinations. Thank you all for taking the time to join this session. I am Martine Perjean, IPOPI President, and it is my pleasure to moderate this webinar. Before we begin, I would like to take a moment to thank CSL Behring for making this webinar possible. Today, we will explore the vaccine recommendations for PID patients. What vaccines are recommended and what are not? And are there new vaccines on the horizon? And especially here with things of SARS-CoV-2. To answer these questions, we have invited a great specialist on the topic, Professor Charlotte Cunningham-Rundles. She is a professor of medicine and pediatrics, a member of the Immunology Institute, and directs the Immunodeficiency Clinic at Mount Sinai Hospital in New York, US. Welcome, Professor Cunningham-Rundles. So, how will this webinar work? Professor Cunningham-Rundles will soon begin her presentation. Once the presentation begins, you can post your questions in the question tab that you can see on the right side of your webinar screen. After the presentation, we will discuss a selection of the questions with both Professor Cunningham-Rundles and with Dr. Nizar Malawi, Chair of IPOPI's Medical Advisory Panel. He will also answer some of your questions in written form. Thank you, Dr. Nizar Malawi, for joining the webinar today. But now let's get started. Professor Cunningham-Rundles, the floor is yours.
Thank you very much, Martine, and especially thank you to IPOPI for making this possible and CSL Behring. I certainly appreciate the opportunity to talk about this topic, although I must confess it's extremely complicated. It's not something which is handled in 1 minute, and so we'll do the best that we can. So let's think about vaccinations in primary immune deficiency. And of course, I've put a number of different types of bacteria and viruses and such on this slide. The idea being to say that we really are only beginning to understand all the challenges that we have in the world of primary immune deficiency. Even though the field has been ongoing for some time, still, I'm sure there's many surprises that we simply, really haven't accommodated ourselves and frankly are still in the learning phase about. So first of all, let's think about what we're going to talk about. I was going to talk about vaccines in general. What is in a vaccine, for example? And then what about people who have a primary immune deficiency? What sorts of vaccines are available? And which ones should we basically be avoiding? And which kind of patients should avoid which kind of vaccination? Then, of course, we want to get to the idea of, well, what's on the horizon? Of course, there's a burning topic right now in the world. And that one I can only touch upon, but we can at least have some opening remarks about that. And the other thing is that I am aware, and I think everyone here is aware, that there is a bit of an anti-vaccine movement. And we can't get away from that fact that it's a problem and we have to deal with it. And it's something that we have to think about very carefully. I just wanted to go backwards for a moment. Vaccination is really something which is not new. This is 225 years approximately now of vaccinations, going through so many physicians and scientists who worked for so extremely long, over such a long period of time, trying very hard to come up with measures to protect against these common bacteria and viruses that you can see on this slide. And as you can see, it's not stopped. Even in 2001, these protein conjugated bacterial vaccines have come along. And as you know, there's other vaccines being invented even now. So this slide, I would run out of room if I tried to put every single invention since 2001. I really like this slide a lot. And the reason why is because this is in Gloucestershire, and it's a small town named Barclay in the west of England. And this is the first vaccine laboratory. And it was Dr. Edward Jenner's location where he was working with cowpox. The reason why I like it is that Mrs. Jenner told her husband there was no way that he was going to be making vaccines in her kitchen. And so she told him he had to find someplace in the garden to do it. And this is actually his hut. And you can go and you can look at that today. It's part of the Jenner Museum in Barclay. I visited there, and to me, it's extremely interesting location. And of course, Obviously, this is the birthplace of vaccination if you get right down to it. Well, what types of vaccines are there? Well, there's live vaccines, and then there's what we call live attenuated, which means that they've been partially killed, but they still have a little bit of ability to stimulate the immune system. And then you have killed and inactivated viruses, and then you have toxoids. I think everyone's familiar with tetanus toxoid, for example. Then you have fractions of cells, which are sometimes used for vaccinations as well. And that would be the bacterial vaccines that we're very accustomed to these days, the pneumococcal, for example. And then we get to some new territory here, recombinant vaccines. Those are going to be new, and we don't know very much about them yet, but the recombinant vaccination started, of course, with hepatitis B, where we have taken a small amount of the DNA and then made a protein which will serve as a vaccination. And then the new ones, the new kids on the block here, the DNA and the RNA vaccines, which, by the way, there aren't any yet, but these are the ones that are currently being explored. So the other thing is you'll notice if you ever had a vaccination, and I'm sure everyone here probably has had more than one, and that is that there's different ways that vaccinations have to be given. It can be given orally, such as the poliovirus or rotavirus, or intramuscularly, and that's this little list here, or just under the skin, or then intradermally. And you might wonder, why on earth do we have to have these things given in different ways? And that's partly because the route of the administration is the way that it's been tested and shown to show the most effectiveness of the vaccination. And so that's the way that we like to give it in the clinics as well. We want to get the best job that we possibly can, and that's why you have to take a look at the method of vaccination. So this is kind of one of the first slides that's just a little bit long-winded in a way. There's a lot to have on this slide, a lot to say, but it again tells you a little bit about what I said a moment ago, which is the virus can be inactivated. Well, how do they do that? They treat it with chemicals, or they can weaken it by the installation of different mutations that weaken the virus. And everything has an advantage, as you can say in the first portion, that first column, but then there's disadvantages. In other words, there's things that are really harder to do. In other words, for virus, the disadvantage is that basically you have to grow a lot of that virus first. And secondarily, for a virus which is alive, obviously you don't want to give that to someone who has a really, really poor immune system, because you could imagine that would be rather, a challenge, too much of a challenge. And the examples are over there on the sides. So each one of these has its own advantages and each one has its own disadvantages. And I think everyone who's been listening to this particular webinar probably is used to some of these. For example, BCG, you know about polio. I think many, many people have had the hepatitis B or the tetanus shots. And then if you look down at that last one, I think that most of the people listening are most likely had what we call the carbohydrate-based, and those are the sugars that are on the outside of bacteria. And that's the way that the human immune system recognizes a bacteria for what it is. It's by the bacterial or the surface coat of that particular organism. And that would be the pneumococcal, for example, and then the Haemophilus vaccination. So in other words, these are some of the methods used. And of course, there's many of them and likely to be more, as I'm going to come to in a moment. So in a way, this is the most complicated slide that I'm going to show. And in a way, it's one where you can't say that all physicians 100% are in agreement. And we can't be absolutely confident that everything that we say on this slide is perfectly accurate. In other words, let's think about that for a moment. So for severe combined immune deficiency, generally, most vaccinations are okay, but you may not get a live vaccination. But there are ones that you should get, which are going to be the flu vaccination, and then the conjugated pneumococcal, and the conjugated Haemophilus. And don't worry, I'm going to come back to that word conjugated in a moment, so just put that on the side. This is where it's a little more tricky, it seems to me, and that in X-linked agammaglobulinemia, the antibody deficiencies, common variable immune deficiencies, we generally suggest that all the generally recommended vaccinations are fine. On the other hand, don't get the live virus vaccinations. But of course, in my experience, many patients with antibody deficiencies have already gotten the MMR vaccination and probably got the chickenpox vaccination when they were children. So that's where there's a little bit of fuzziness in the language here. And that is that most of those vaccinations might be okay for these patients, but there are some that we really are very unhappy about. For example, the oral polio, the live polio, is really one that caused quite a lot of devastation for the patients with X-linked agammaglobulinemia. And then looking on down, then you have people, children, for example, and adults who have T-cell deficiency, also DiGeorge syndrome. And again, live viruses are contraindicated. On the other hand, after a child has exhibited some evidence of immunity, most of us will then go ahead and allow that child to have certain vaccinations. Again, that's going to be at the discretion of the physician and also looking at the individual patient. Looking down the slide, the complement and the phagocytic defects, that's going to be chronic granulomatous disease. Again, all the vaccinations are generally going to be reasonable. What about if you have no spleen? That's a very specific category. And again, all the generally recommended vaccinations, but we tend to not worry very much about the live viruses there. But we do suggest very strongly taking the conjugated vaccinations to protect against bacterial infections. So you might wonder in your own life, what are the live viruses? And that is these here: measles, mumps, and rubella, the rotavirus, chickenpox vaccination, and the older version of the zoster vaccination. We do now have a killed version of the zoster vaccination. I'm not certain how widespread that is, but that one is a very successful substitute for the zoster vaccination. Of course, the BCG, Japanese encephalitis, oral typhoid, yellow fever, and then in the oral polio. Now, we haven't had the oral polio vaccination in the United States since the year 2000, but it does exist in other countries. And of course, we also have a specific recommendation for household contacts, and that is going to be this group of vaccinations as well. Again, you could wonder whether this is safe for a family in which one child is extremely immune deficient, and then household contacts would then receive these vaccinations. And this is again where I think some discretion has to be involved in order to really be absolutely sure that we are giving the right advice. I wanted to go back for a moment into the issue of, I used the word conjugated vaccines. What is that anyhow? So what this is a way of making the antibody in gamma globulin production bind more effectively. And to do that, one way to do that is you attach to the sugar from the bacteria a certain amount of protein. So the B cell down there, this happy little B cell, is going to recognize the one that's containing this extra bit of protein in order to make it more immunogenic. Sugars themselves are not, unfortunately, very immunogenic, but those are really on the surface of all bacteria. That's the way bacteria are actually surface constructed. And so really a good way to make them a little bit more immunogenic is to add a protein on the surface. So you can see that happy B cell down there now is making antibody. So the question is, what is that protein anyway? Well, fortunately, that protein is one that we actually want an antibody to anyway. That will be diphtheria, for example, or tetanus. One component of the meningococcal outer membrane complex, or diphtheria, or one of the other major proteins from influenza D. So it's another way of making the immune system see it a little bit more vigorously and therefore make a better antibody response. I think probably most of the people who've listened to this particular webinar are very familiar and probably have already received a conjugated vaccine, but that's what's conjugated about it. It means you're making the immune system pay more attention by hooking onto a protein that the immune system sees a little bit more vigorously. Now, of course, many people who are on this webinar and are listening will say, well, you know something, I'm on gamma globulin, so what vaccines do I need? And that's again tricky, because if you're on large doses and good doses of gamma globulin, this has been examined quite a number of times. And if your lowest IgG level is well within the normal range, you will have protective levels of antibodies to these issues here, to Haemophilus, measles, mumps, rubella, and diphtheria. But a lot of studies have noticed that for tetanus and for some of the pneumococcal bacteria, the antibodies in gamma globulin can vary. And in those circumstances, for a person who can make some degree or a little bit of immunoglobulin, getting an extra dose of a vaccination is never really a bad idea. Now let's talk about something just a little bit more complicated, which is this. Now, this is the way the immune system looks. If you simply take a big picture and you say, what are the components? Well, you have stem cells that then turn into lymphoid stem cells. And then those are the ones that ultimately either in their lifespan are caused to slowly become and make up their mind. Their job description is to be a B cell. And B cells, of course, you've heard that phrase probably many, many times. B cells then mature and then produce what we call plasma cells. And plasma cells live in the bone marrow and the spleen and other lymphoid organs and produce large amounts of gamma globulin. So you can see those are the gamma globulin molecules over on the side. And this is what vaccinations primarily are working upon. They're working on these cells, picking up antibody information, which then turns into circulating IgG, which then will bind to the relevant bacteria. And so that's this little set of cells here. But there's one other thing to say, and that is that the other section of the immune system, which are the T cell side, these cells actually have their own tricks, and these cells do not produce gamma globulin, but what they do is they produce things such as gamma interferon and interleukin 2, and these are also quite important for immunization. This is a study that I took out of the literature. It's from German investigators in Berlin, actually. It's a very pretty study. And you might wonder, when you have a person who does not make immunoglobulin, why are we suggesting flu shots? We're not asking that person to make an immunoglobulin, but what we are saying is that the T cells can pick up information as well. And this has been proven, and that's why I like this study, and that is that you can see that the hypogammaglobulinic patients, that one in the first panel produced a tumor necrosis factor, which is a kind of a cytokine that helps fight infections, and interleukin 2, again, the same story. Perhaps it's not as much as the normal control, but it's certainly not negligible. And look over on the interferon. The interferon was really just the same as the normal controls over there, really very substantial amounts of interferon. So this is why even if you don't make antibodies, and perhaps you're even on immunoglobulin solution for that very, very reason, this is why your physician is suggesting that you do get a flu vaccination. And I know that we do that, and I'm sure most of the clinical immunologists that would be, you would go to see would say the same. Now this gets tricky, of course, because now we need to know what about new vaccinations and what about the SARS vaccination and how is that going to affect our immune deficient patients? And that's tricky. Of course, we need to stop this virus. We need to say, please, this is enough of you. We need to, over time, develop some method of dealing, and ultimately, of course, antibodies to this particular virus will be present in the gamma globulin solutions. But unfortunately, that's going to take time. To my patients, I generally say it's going to be probably a year and a half or more, just because blood donors are slowly going to be giving blood, and over time that antibody will appear. But what about in the meantime? Now, if you notice that this was the slide that I showed a moment ago, which said these are the types of vaccinations, but the new ones down on the bottom in the red, nucleic acid made by gene methods, are going to be either RNA or DNA. And you might say, well, what is the purpose of that? These are RNA or DNA that are extracted from the virus genome, in other words, from the DNA of the virus, which then inserted into a human cell will cause the human cell to make a certain amount of the proteins that will stimulate an immune response. So it will be dead. The DNA or the mRNA, the messenger RNA, will be dead. They will not be alive. They will not be able to replicate. On the other hand, the immune system will be turned into a tiny little factory producing a certain amount of these proteins so that the immune system can then go ahead and make an immune response. At the moment, these are considered safe because, of course, they don't contain live materials. They're easy to develop. But what are the examples? Well, there aren't any. We don't know yet. That's the honest truth. We don't know yet. We don't know what the difficulties will be. We don't know if it'll work. And we also don't know if it will last. Another point about the immune system, of course, is that you not only want to have immunity for the next month or 2 months or say half year, what you would like to do is develop an immune response that can last for several years, just as other vaccinations do. And that's where, of course, we don't know what the answer will be, but these are new. So we shall see. So as you probably have heard, and this has probably changed since I made the slide only a few days ago, there's 120 candidates for this vaccine in development, and they were begun within the first 5 months of 2020. And they are composed of a lot of different techniques. As you could imagine, there's a lot of ingenuity that's going into this currently. Inactivated virus, as I said a while ago, is a way of just killing the virus, but making it still capable of inciting an immune response. Maybe you could just take off some of the viral protein, like the spike protein, the surface protein of that virus, and use that as a vaccination, as we do, for example, tetanus or for diphtheria. And then these, of course, are the DNA vaccinations, as you can see, that will contain the DNA for the SARS virus spike protein. And then the host cell was supposed to read the DNA and convert that into that spike protein. And then subsequently, that should result in the production of both antibodies and killer T cells. Do we know that? No, we don't. But there's data in many, many models that says it should work, and it perhaps even would work. It's just that we can't say this is settled yet. Another one is taking a very harmless adenovirus, which is a cold virus, and putting the spike protein on the surface of that cell. So the immune system is a little bit tricked, thinks it's the SARS virus, but actually it's not. It's just a cold virus. So the immune system sees that as foreign, and then antibody is made. And the last one is one of the most interesting and curious of all, which is the messenger RNA, which is kind of an instruction kit for making the spike protein. And then the idea is that in the immune cell, that material is made, and then it's then perceived as a vaccination on its own, and then antibody is made. Now, how do we know that any of these will work? Well, I'm not going to say that we do. I think clinical trials have shown that there's reasons for hope, and I do think that we will have probably more than one successful vaccination, but as you know, this is going to have to resolve over time. This is a slide, I don't think you can see this, but the point is that there's a lot of ingenuity going into this particular methodology. And if you go online, you can find this same slide too. It's slide 1 and slide 2, saying how much effort is ongoing. This is not only at medical centers, this is not only amongst scientists at research institutions, but of course, it's one that many, many companies are very, very interested in developing a vaccination as well. There's a lot of governmental push, of course, naturally, because you can see this has been very, very difficult. Now, you might just quickly like to know, how is success determined? How do you know if you have a good vaccination? Well, you're going to have to find new antibody in the blood that you didn't see before. You're going to have that antibody kill and bind and then kill that virus. So that's called neutralization. And the other thing, which I alluded to a moment ago, is that ideally, the other section of the immune system, the T cells, would slowly develop immune protection as well. It's not just an antibody phenomenon. We like the T cells to gain knowledge too. And the last point is that all types of virus must persist. In other words, there's no use to have something which will last for a few months and then disappear. That would be really quite useless. And this is what it would look like. You see, this is what we call titer, by the way, if you've ever heard your clinical immunologist use that word, which is that when you start, this is just a clinical trial that was used on one of the many vaccinations, and it was published pretty recently in one of the journals, which is that this person is starting on several different, this is a group of patients rather, given either 1 or 2 or 3 doses of this particular viral protein. And they start off with very little antibody. That's why it's down at the bottom, that low number. And then over time, over the next, say, 57 or 60 days or so, then antibody is then detected in the blood, and hopefully it's useful antibody that can kill the virus. And that's another picture that I didn't give you, but this is what's called titer. Titer simply means how much can you dilute that serum and still see something which is still positive or binding. And that, of course, is an indicator of how much antibody is actually present. So will this be safe? Well, the jury is out, but with no live viral elements, it should be safe. But would there be unintended consequences? We can't say that there won't be. We don't know that. And so this is where the jury is out. It could be some random side effect that we have not thought about yet that will perhaps develop, and we won't know that. So I know that immune deficient patients will probably not be the first to be immunized. It will be healthy volunteers with the normal immune system. And then, of course, the next thing is, even if you are immune deficient and you get a vaccination and it was safe, would it work? Will there be any antibody? Well, that's uncertain, isn't it? I've had some of my patients be infected with the SARS virus, and they, in fact, did make antibody, which I can now detect. And so it certainly depends upon the type of immune deficient and the depth of the immune deficient situation. And then, of course, the other thing is that we'd really like to know, does it lead to that T-cell immunity? Ideally, yes, but again, that will have to be determined, and it's not known currently. Now, what about the anti-vaccination movement and its implication for our patients? Well, what's with the anti-vaccination movement and its implication anyway? First of all, of course, would you get a vaccination yourself? Would you like to get a vaccination? No. I mean, actually, no one probably likes vaccination, but one does like to be safe, and one does like to go out in society and conduct your profession and travel and basically be with one's family and one's friends. And so, no, we wouldn't do it for pleasure. That's number one, but we would like to have us have all those advantages. And then, of course, there are rare adverse events. We can't get around that fact. Even to every vaccination you've ever seen, the package insert will tell you that. The next thing, of course, is the content is a viral or a bacterial product. And who likes the idea of being injected with a viral or bacterial product? We can't possibly like that idea. It's just that, as I say, we do like to be able to conduct our lives with great freedom. And this is something which then allows that. And then question, of course, about the additional contents is something which always comes up. Of course, there are vaccine reactions, and I've just copied this from, I think this is the CDC, the Centers for Disease Control in the United States. And as you can see, every vaccination has negativity. Most of them are going to be under very, very specific situations, such as the live virus polio vaccination has a very old history of being a difficulty for patients with X-linked agammaglobulinemia or those who have no circulating B cells for other reasons. And then, of course, measles, perhaps. You can look down the list of things that have been cited. And this is all in the package insert. And we, of course, we can't get around the fact that these do occur. Is it common? Not particularly. By the way, I would not say it was more common amongst immune deficient patients than it was amongst normal, everyday, immunocompetent individuals. The other thing is about a vaccination, you know, it's not just the viral or the bacterial protein. There are additional contents, naturally. In those multi-dose vials, mercury was added in the past as a preservative, and now it's only in some vaccinations for flu virus. And actually, you can get it mercury-free. Formaldehyde, which is a preservative, again, for multi-dose vials. Most physicians don't use multi-dose vials. They use the single one by one, so that would, again, not be needed. Aluminum has been in some vaccinations because it makes the vaccination stronger. There could be a trace of antibiotics. There can be a trace of gelatin. And in some vaccination, there's a trace of monosodium glutamate, which is a preserver for tiny amounts, but it's only present in tiny amounts, as you would imagine. And so it's not just the one thing. It is containing very, very tiny traces of additional materials. And I wouldn't say we're worried about any of these. The only one that people have thought about a lot is mercury. And measles, mumps, rubella don't contain mercury, actually. That's the thimerosal. That's what the mercury comes in that form. And as you can see, others of these vaccinations do not contain that material, and flu vaccinations are available in both situations. The issue about mercury was always for the issues of autism, as you know, which was also actually discarded as a scientific idea many, many years ago. And so we actually don't consider this as a problem, but as you can see, mercury has slowly disappeared from vaccines in any case. So having said some of the negatives, you just cannot possibly get around the benefits for immune deficiency. And for all of us who treat patients, and for all of you who have an immune deficiency, or any of your friends who have an immune deficiency, you just have to say that a vaccination is one of the most cost-effective, life-saving medical interventions that has developed. I can almost not think of anything else which has been going to be as cost-effective and life-saving as this particular measures. And of course, it's been estimated to save many, many, many lives per year. And then for an immune deficient person, safe vaccines then allow one to develop this degree of protection, which then allows you to conduct your life, go to weddings, funerals, restaurants, and basically, as some of my patients are physicians, for example. And why would that not be an agreeable situation to choose your profession based on your knowing that you are protected? And then, of course, when close contacts are vaccination, further protection is provided. I just copied this slide and put it out. I just think this is an incredibly beautiful slide that someone put together the data about this. This is vaccinations and infections over 1 century. I show this to our medical students at Mount Sinai because we tend to take antibodies for granted. We tend to think, oh, well, it's fine. But, it wasn't fine, for example, 100, 200 years ago, it wasn't fine. Diphtheria, for example, tetanus, whooping cough, for example, measles, for example, scarlet fever, typhoid, all of these things were rampantly causing shortening of life during previous times. The Australians also put together their data for vaccinations. Again, I think it's a labor of love to have put together this particular paper showing the number of deaths in Australia from diseases vaccinated between 1926 and 2005. And to me, I just think this says everything, which is that vaccinations has really been something which has been a marvel. I think for everyone on immune globulin 2, you are taking advantage of immunized donors. You are taking advantage of all of the people who now are supplying you with this antibody as well. Again, very, very, very perfect. So anyway, thank you very much for asking me to talk today. And I have to say thanks to all our amazing patients and our wonderful nurses. I have some fantastic medical assistants, the Immune Deficiency Foundation and the Jeffrey Modell Foundation in the United States, and most especially to IPOPI, which has made this international organization so wonderful. So thank you very much, Martine.
Thank you very much, Professor Cunningham-Rundles, for this interesting, brilliant presentation, for putting all those information together, because it's a huge amount of information, and I think it's really, really important and interesting. So thank you very much. Welcome, Nizar. I'm very happy to have you with us. So we have many questions, and I propose to have 2 times. First of all, we will begin with a general question, and then we will move to COVID-19 vaccines, because we have the 2 of them. The first one I would like to ask you, let me see, this is what happens if a mother or father has a PID and their children need to start the vaccines such as MMR. In some countries, kids are not allowed to go to school unless they are vaccinated. Should the parents isolate them for 48 hours?
I just knew you were going to ask me the hardest one first. There's really no consensus, but what we would like to do is to have our children go to school, that's for sure. And we would like to have our children immunized. So if you have an extremely immune deficient adult who's perhaps been bone marrow transplanted or has an extremely severe T-cell deficiency, isolation for several days is not a bad idea. I have never seen anyone do any study on that to know how the risk actually is. Whether someone could show me a paper that tells me how long that period of isolation should occur. I've never seen good data on that, frankly. I've never seen that situation played out in real life. In other words, the person developing any of these illnesses in the face of an immunized child. I am sure it's existing, but I don't know the literature on how common that is or what one would do about it.
So case by case. I'm afraid you will have some tricky questions, but well, let's go. Another one from India. This patient received measles vaccine before he was referred to us. But if a patient is diagnosed to be combined immunodeficiency and has not yet received measles vaccine, would you recommend give this vaccine?
Is this individual going to go on to gamma globulin treatment?
Okay.
So to me, on immunoglobulin treatment, the need for doing that immediately is lessened because you are going to be protected against measles, mumps, and rubella by the immunoglobulin. And so there's less of a rush. There's no reason to push that immediately.
Okay. Still from India, we have a patient with combined immunodeficiency, not severe combined, developed fatal measles, including body encephalitis, likely vaccine-induced, as there was no history of measles infections. I think this is the complement of the question, something like this, I guess. So let's move to another kind of question. We have a question on vaccines and HSCT. Do you give a vaccination for PID patient before HSCT? And the other question is, what about vaccination after HSCT?
Well, generally, a person who's having a bone marrow transplant perhaps is already immunized, or it's severe combined immune deficiency, in which case you would do it afterwards. In other words, if it's an infant, you would do it afterward, hopefully after the graft is already taken, and then that new cells can then pick up that education. But if it's an older person, our policy here is to then go through the entire vaccination routine somewhere after engraftment has occurred, 6 to 9 to a year later. And so usually all immunizations have to be repeated after a transplant.
Yeah. Okay, Nisa, if you want to add, I let it to you just.
Yeah, no, I should totally agree. Maybe just for the post-transplant question, I guess one might want to, want to consider the quality of the immune reconstitution, and especially the T cell population, because then if it's a fully reconstituted graft and immunity, well, that's fine, and you can really go through all the the live vaccines as well, but if there is still a little bit of low T cell, and again it depends on how low it is, then one might be careful. But usually clinical immunologists know very much about that, as well as the bone marrow transplantation team.
In the meantime, the immunoglobulin will bridge the gap, and hopefully the engraftment will occur. And it will be satisfactory, but you wouldn't use a live virus in someone whose T cells are looking pretty weak.
Yeah, absolutely. Another question: I have concerns about autoimmune events in susceptible subjects as CD40L or complement deficits. Would you comment on that?
I'm sorry.
So someone says he has or she has concerns about autoimmune events in susceptible subjects like CD40L or complement deficits. Would you comment on that?
Vaccinations in CD40 ligand or complement deficiency.
Yeah, absolutely.
Right. And the risk of autoimmunity events after the immunization.
Yeah, exactly. This is a question.
So there is a certain risk about particularly ITP, I think, low platelet counts, that was on that slide. You can see that that's listed in the package insert for measles, mumps, and rubella as being something to consider. You you're going to have to weigh one against the other, one circumstance against the other. A person with CD40 ligand, again, is likely to be on immunoglobulin solution, most likely. So you might have a person who you need to get into a very stable situation and then perhaps, immunize. But I don't feel terribly strongly about immunizing with measles, mumps, and rubella in a person who is on immunoglobulin in the first place. So the problem would perhaps be someone who's complement deficient, who probably is not on gamma globulin. This is the same risk people with previous history of ITP face. For example, CVID, or even just anyone with previous history of low platelet counts, giving an MMR can, and it has been known to perhaps lower the platelet count. It's rare, but it does occur. So you have to weigh one thing against the other there.
Yeah, thank you. We have many questions coming, so I will choose one which will allow us to bridge to the second part. What about mutation of virus and vaccines? Because we know that, for example, the flu vaccine, the flu virus mutates. So how do we deal with that, and especially in the perspective now of the SARS-CoV-2?
Well, I of course give all my immune deficient patients the flu shot. The best you can do is to give the latest version of the flu shot, realizing that we always have to make the guess in the summer as to which one is going to be rampant. And I don't think we get it right every single time. That's just unfortunately true, isn't it? So there's no wonderful answer to that particular question. We have to, use the vaccination as it stands, which of course is no risk to a patient with any of our immune deficiencies because it is not alive.
Yeah. Okay. So going to COVID-19, I have a question about, you spoke about the anti-vax. So what about the potential rumored auto-inflammatory side effect concerning the new RNA vaccines in the context of immunodeficiencies and crossover autoimmunity and auto-inflammatory?
There's no good answers to that at the moment. There's so many lessons the immune system has to teach us that I'm not going to be able to tell you that that's clarified as to cause. For every vaccination ever invented, there have been some unexpected events. If you look at the physician's desk reference, you'll see that there's adverse events for every single vaccination ever created, even those that have been around for, I don't know, say 5 decades or so. So will we see the same thing with these COVID vaccinations? I think we will probably do exactly that. I think we will see additional side effects that we didn't dream of. Is it likely? It will, again, be a rare event, but we can't say it's not going to occur because for every vaccination there have been deleterious effects.
In some countries, we will have together flu and COVID-19 coming or coming back, I don't know. And what would you advise when it comes to vaccines? And would the flu vaccines protect you against COVID-19?
Well, the flu vaccine won't, because everything in the immune system is, one by one. You learn one thing, like the multiplication table doesn't teach you the alphabet. Do you know what I mean? So it's kind of like one thing at a time. I have actually been telling my patients, don't take the flu vaccine until there is a little bit later, because we haven't got any flu yet in the United States. And you like the peak of your immune response to be a little bit later. And so I know there's a great push to go get the flu vaccine right now, but it's actually, in my view, premature. And there is data that says that since your flu vaccination T-cell recognition is maximum, at a certain interval, you would rather have that match the time when flu is around. So I'm not suggesting flu this very second. I'm saying in somewhere around 3 to 4 weeks. I think it makes more sense. I don't know, Nazir, what do you think?
No, yeah, I agree. I agree with you. Yeah.
I think people are afraid that once you say postpone, they'll forget to do it. Don't you think? I think that's the problem.
Okay. Always still from the patient perspective, we know now that there are many vaccines, possible vaccines. how will we have a possibility to choose, and on what criteria? And because we understand from your presentation that some will be different from the others, of course, and it will be tricky, won't it?
It'll be very tricky.
Yeah.
I mean, I think, you know, all of the scientific and patient organizations are going to have to come together to come up with the best and most logical types without making the attempt to endorse a company, for example, or an endeavor, but we will end up having to have recommendations based on the data.
Absolutely.
There's no doubt about it. We're going to have to get together as a world committee and, you know, come to our conclusions. And I suspect there'll be several candidates that will be very, very workable, but we have to have a consensus on these things.
Yeah, I will maybe take one or two more questions, and I'm going back to the general. There are countries like Romania where there is no newborn screening for PIDs, and children receive BCG vaccination in the first day of life, and it's not only in Romania, I can tell. I know this can be catastrophic for skilled children. What about, how risky for other conditions, other immunodeficiencies?
Well, this is a problem that's existed forever, right? And we've learned the hard way about BCG, for example.
Yeah.
And there's no other, you know, result that we can think of. In the United States, we don't have the live flu flu oral polio anymore, even though the risk was tiny and it probably avoided flu for so many people. There were patients who bore the cost of that. But that's unfortunately one of those things that we're going to need to learn. I think with the particular COVID vaccinations, the ones that seem to be coming along are dead. They are inactivated, they are not alive. And they may have their own unusual or strange side effects that science has not learned yet, but most of them are basically going to be dead. There is the exception of the cold virus one, which is a cold virus actually, and I would suppose that would be one that we would not choose.
A very last question coming from Iran. What about vaccination in children diagnosed with having LADs?
LAD.
Yeah, LAD. Sorry.
Leukocyte division deficiency.
Leukocyte adhesion. It's a very rare ailment, and I haven't heard of vaccination difficulties in particular, but of course, local infections are very, very common. So I think local reactions could actually be rather more tricky there. Live viruses in most situations might not be a particular negative, but I think you would want that child to be perhaps, you know, not an infant at that stage so that you can follow carefully. Which vaccination is being considered under the circumstance?
It's not indicated.
Well, maybe one can add that as for SCIDs, LADs are usually diagnosed during infancy. Not all of them, because sometimes you can have a more like delayed diagnosis of a milder form. But then these patients should undergo bone marrow transplantation, and then we come back to the discussion regarding vaccines and PTSD.
Right.
So I think that we will stop right now to keep in time. And so we have reached now the end of this webinar, which was made possible thanks a kind donation from CSL Behring. Thank you for that. On behalf of IPOPI, I would like to say a warm and huge thank you to Professor Cunningham-Rundles for this excellent presentation, amazing, and for all the answers you're providing, and also to Dr. Nizar Malawi as well for your great contribution. Thanks again to all of you for attending this IPOPI webinar. If you have a minute to spare, we encourage you to share your feedback on this webinar by pressing the link that you can see right now on your screen. This link will also be posted in the chat. Please note that you will be able to see the recording on IPOPI TV within a couple of days, and we encourage you to share it as much as possible. And see you next time with a great pleasure. keep her healthy, and see you next time. Bye-bye.
Bye-bye.
And thank you again.